Hello and welcome to Diversity of Science. Today's video is going to be about discussing the first principle of pharmacokinetics, which is absorption. We will learn how absorption is carried out inside the body and the different mechanisms that carry out this absorption, like passive transport, active transport, endocytosis, and endocytosis. So without wasting any further time, let's begin. Pharmacokinetics. What is pharmacokinetics? Pharmacokinetic refers to what body does to a drug when it is inside the human body. It determines the intensity, duration of action, and the onset of the drug. It works on four principles: A, D, M, and E. A refers to absorption, D for distribution, M for metabolism, and E for excretion. Now, as I said, this video is about absorption of drug. Absorption is the transfer of drug from the site of administration to the bloodstream. Now, this is carried out in three mechanisms: passive transport, active transport, endo and exocytosis. Let's discuss passive transport. Passive transport is the movement of molecules from a region of higher concentration to lower concentration. Now, this is carried out in two ways. Number one, by simple diffusion, which means it does not require any helper protein, and number two, by facilitated diffusion, which means it will require helper protein. Now here you can see that in simple diffusion, there are diffusing particles outside of the cell and they are moving inside of the cell without any carrier or helper proteins across cell membrane. The example of this type of diffusion is the exchange of gases in lungs and blood. The other type of diffusion we have here is facilitated diffusion. This type of diffusion occurs using a helper protein. Now there are two sorts of helper proteins. Number one, channel protein, and the number two is carrier protein. Both of these proteins entrap the drug molecules inside of them and released across the other side of the cell membrane. The example of this type of diffusion is the movement of glucose molecules. Another type of transport we have here is active transport. Now this is the movement of molecules from the lower concentration to higher concentration, which is the opposite of passive transport. The example of this type of transport is sodium-potassium pump, which helps to move potassium into the cell and carry sodium out. From the picture, you can see that the movement of molecules from lower concentration to higher concentration is occurring by the conversion of ATP into ADP and the release of phosphatase ion. ATP is adenosine triphosphate, while ADP is adenosine diphosphate. Another type of absorption is through endocytosis. In this process, the foreign particle is engulfed within the cell membrane by forming a vesicle, which is then later destroyed by a cell organelle, which is lysozyme, and the contents are released inside the cell membrane. Now, this is further carried out by three processes: phagocytosis, which is cell-eating process, pinocytosis, and receptor-mediated endocytosis. Now looking at phagocytosis, you can see that the large particle is engulfed within the cell membrane by forming a vacuole or a vesicle. The example of this is immune cells such as white blood cells take up viruses or bacteria, digest their contents, and move them outside of the cell. Another type of movement is pinocytosis, which is also called as cell drinking process. In this process, small particles, fluids, and dissolved nutrients are taken up within the cell. Once inside, it combines with the lysosome, which degrade the vesicle lining and release the molecules into the cell. Example of this type is absorption of fat from the cells of small intestine. You can see that the small particles of fluid which are outside of the cell enter into the cell through a similar process like phagocytosis. Now the last type of endocytosis we have here is receptor mediated. This is used by all cells to take up specific molecules by the help of membrane receptors coated with a special type of protein called clathrin, which traps the substance by forming a vesicle. The example of this type of endocytosis is the uptake of cholesterol in the form of low-density lipoprotein, LDL, inside the cell. From the picture, you can see that the small particles of low-density lipoprotein are swimming in the extracellular fluid. Now, the receptor on the plasma membrane entrap these particles. They form a vesicle, which is then later released inside of the cell. The last type of transport we have here is exocytosis. Now, this is the process in which the molecules are moved outside of the cell, since exo means out. It occurs when the vesicle fuses with the cell membrane, allowing its contents to be released outside of the cell. Example of this type of movement is neurotransmitters such as norepinephrine, cellular waste, sweat, and secretion of glands such as insulin. Now from the picture you can see that a newly formed vesicle that contains waste is allowed to fuse with the cell membrane, which then later releases its waste outside of the cell. This was all from my side for today. Thank you so much for watching. Hope you guys understood what I've taught you so far. Take care and happy learning.